Hello, everyone, and welcome to today's live broadcast, Companion Diagnostic and Biomarker Assay Development for Interrogation of Liquid and Solid Tumor Biopsies from RNAscope and Exosomes. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Advanced Cell Diagnostics. For more information about our sponsor, please visit their site at acdbio.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd now like to introduce our presenters. Dr. Robert Monroe, Chief Medical Officer, Bio-Techne, Dr. Christopher Bunker, Senior Director, Business Development, Advanced Cell Diagnostics, a Bio-Techne Brand, and Dr. Michael Valentino, Scientific Director of Companion Diagnostics within the Business Development Team at Exosome Diagnostics, a Bio-Techne Brand. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Dr. Monroe, you may now begin your presentation.
Welcome to today's webinar on the development of biomarker and companion diagnostic assays from RNAscope to exosomes. I'm Rob Monroe, Chief Medical Officer of Bio-Techne, and I'll be joined by two of my colleagues, Dr. Michael Valentino, who is the Scientific Director of Companion Diagnostics at Exosome Diagnostics, and Dr. Chris Bunker, Senior Director of Business Development at Advanced Cell Diagnostics, known to many of you as ACD. Both Exosome Diagnostics and ACD are Bio-Techne brands, and I'll be telling you a bit more about Bio-Techne in the next few slides. Our goal for today's webinar is to provide you with an overview of the Bio-Techne technologies that are currently being used by biopharmaceutical companies for the development of biomarker and companion diagnostic assays for their specific therapeutic programs. We plan to highlight the use of exosomes and RNAscope, RNA in situ hybridization as companion diagnostic platforms for biomarker assessment and patient selection, with a specific focus on oncology applications. Before I pass the baton to Michael and Chris for a more in-depth discussion of these technologies, I'd like to first give you an overview of Bio-Techne, since many people are not as familiar with our parent company as they are with some of the individual Bio-Techne brands, such as R&D Systems and Protein Simple. So what is Bio-Techne? Bio-Techne is a Minneapolis-based life sciences company with over 2,000 employees spread across 35 sites around the world. The company manufactures and distributes a very broad range of consumables that include antibodies, proteins, immunoassays, small molecules, and in situ hybridization reagents, among other research tools. As highlighted in the pie chart, the consumables business comprises the majority of the company's sales, but is also complemented by a growing instrument business, specifically for automated protein analysis, as well as a services business that's focused on the development of assays for biopharma and diagnostic industry partners. By the numbers, Bio-Techne's brands produce 300,000 products and 6 instrument platforms that are backed by 42 years of manufacturing experience and over 600,000 publications. Bio-Techne's core products include antibodies, proteins, immunoassays, small molecules, RNA-ish probes, and exosome tools. These products support the company's participation in a number of exciting areas of the life sciences and diagnostic markets, highlighted in this slide, including cell culture and gene therapy, tissue biopsy, liquid biopsy, and automated protein analysis. The focus for today's presentation are the technologies that support the tissue and liquid biopsy approaches for biomarker and companion diagnostic assays, including the RNAscope and exosome analysis platforms. In thinking about biomarker and companion diagnostic assays, I think that it's helpful to look at this testing in the context of a cancer patient's diagnostic journey. Typically, this journey begins with a screening test. For cancer, the most common screening tests are mammography and colonoscopy, but They can also include traditional serum immunoassay-based liquid biopsy tests such as PSA, CEA, and CA-125, as well as emerging highly sensitive approaches that examine cell-free DNA, circulating tumor cells, and extracellular vesicles such as exosomes to look for evidence of cancer. Once a cancer is suspected, the patient typically undergoes a biopsy or surgical excision for a more definitive diagnosis. The resulting tissue is used not only for diagnosis of tumor type, but also for further characterization with prognostic and predictive tests that provide information on a tumor's expected behavior and suitability for specific therapies. These latter assays that predict whether a patient's tumor will be responsive to a given therapeutic, as you know, are called companion diagnostics. Following diagnosis and patient stratification, patients typically will be monitored going forward for evidence of response to therapy, as well as potential recurrence with various approaches including liquid biopsy. Similar to the liquid biopsy test used for screening, the monitoring tests also leverage cell-free DNA, circulating tumor cells, and extracellular vesicles to look for evidence of recurrent cancer, along with genomic alterations, including those that confer resistance to given therapies. Exosome Diagnostics and ACD have developed technologies to support assay development at these three phases of the cancer patient's diagnostic journey. For example, in the screening and monitoring phases, Exosome Diagnostics' exosome analysis platform provides for a rich source of transcriptomic, genomic, and proteomic information for detection of low abundance mutations and expression of specific RNA targets. In the subsequent diagnosis and stratification phase, ACD's RNAscope platform provides highly sensitive and specific technology for the assessment of biomarkers in their spatial context. This allows the researcher or pathologist to correlate biomarker expression with cellular morphology, which is a major advantage over traditional grind and bind molecular assays. In this slide, a potential use of exosomes and RNAscope in the diagnostic journey of a hypothetical lung cancer patient is highlighted. Initially, the patient is identified as having a suspicious lung nodule by CT scan and undergoes an exosome-based lung cancer assay for EGFR mutations on a blood sample. The exosome analysis leads to the identification of an EGFR L858R mutation, as shown on the left. The patient subsequently undergoes a lung excisional biopsy to remove the tumor for definitive diagnosis and mutation profiling. Following classification as a lung adenocarcinoma, the tumor is tested with an RNAscope technology called BASESCOPE for the presence of the EGFR L858R mutation. As you can see in the middle panel, the tissue-based assay is positive for the mutation and correlates with the exosome analysis. As a result of the identification of this mutation, the patient is subsequently treated with the targeted EGFR therapy, erlotinib, which is an EGFR inhibitor shown to be effective in lung cancer patients with this particular mutation. At some time following treatment with erlotinib, the patient undergoes a liquid biopsy for monitoring purposes, in this case for evidence of recurrence in the development of resistance mutations. The exosome-based test shows the presence of an EGFR T790M mutation in the right-hand panel in association with the original L858R mutation, indicating that the patient has developed resistance to the erlotinib treatment. This resistance mutation is also confirmed in a follow-up CT-guided lung biopsy, which shows the presence of both the original driver mutation in blue and the new resistance mutation in red with the BASESCOPE assay. Hopefully, this example highlights how these technologies can support testing across the cancer patient's diagnostic journey. And with this overview in hand, I'd like to hand over the presentation to Michael for a more detailed look into exosomes.
Thank you, Dr. Monroe, for that introduction, and thank you everybody for joining me today. Very excited to share with you how we at Bio-Techne and Exosome Diagnostics in particular are leveraging exosomes for the development of biomarker and companion diagnostic assays. So by way of introduction, liquid biopsy and non-invasive biopsies have been around for a number of years and kind of exosomes are among the newest players in that field. Probably you should be well familiar with modalities like cfDNA and circulating tumor cells. And there are some pros and cons to these different modalities. And so what gets us really excited about exosomes and why they're really a growing force in the liquid biopsy field is because they provide a rich source of information from that patient, encapsulating the transcriptome and proteome of that individual. And it provides detection of low abundance RNAs because it's also more abundant and more easily manipulatable in terms of clinical trial support than these other modalities like cfDNA and CTCs. And something that excites us at Exosome, as well as our partners, is because it gives you the active snapshot of these processes that are happening in the patients that you're interested in. And so just a little bit about who we are at Exosome Diagnostics. There are two commercial divisions. I'll mention, but won't speak to very much, our diagnostics division, where we have launched our flagship diagnostic known as ExoDx Prostate IntelliScore, or EPI. It's actually the world's first exosome-based liquid biopsy. This one happens to be based in urine as the biofluid, and it's a 3-gene RNA signature that is used for risk stratification of high-grade prostate cancer patients. And very exciting for us. We've been very active and great uptake along with reimbursement like Medicare and recommendation in the National Comprehensive Cancer Network guidelines. And so this is really exciting for us, and for the patients that we're helping along the way. However, will speak to our biomarker services, our CDX division, which is where I'm the scientific director in that team. And so what we do for those services are customized projects based on your clinical trial needs and focuses. And so we're very bespoke, which is advantageous to all our partners because you can really customize and focus on the things that you need for your programs. And we span kind of any stage along those programs, from early biomarker discovery, which I'll spend some slides talking about next, into translational assays, and then designing, developing, and supporting clinical assays as well. So just a little bit about exosome biology. As mentioned, exosomes will capture the active transcriptome and proteome of those patients. And so why that's really great and how that differentiates us from other modalities like cfDNA is because exosomes are released by all living cells into biofluids. And so that's health and disease. And so you can really listen in on the signals and the communication that are happening within those patients. And they play roles in this cellular communication in health and disease settings. And while exosomes are really small and they carry snapshots of what that parental cell is communicating with, when you take them together, it actually provides a very big message. And so we do a lot of work in RNA and protein, and for the sake of time today, we'll focus on the RNA work that we do. And I'm very excited to share with you our biomarker discovery engine, which is built on a long RNA-seq pipeline analysis. And why we've chosen to focus on long RNAs is probably if you're familiar with exosome biology, the early RNA work was all largely focused on small RNAs, largely technical limitations and not biological limitations. And we chose to focus on designing and developing from scratch a very sensitive and powerful platform to get at these diverse long RNAs, with the mindset being that many of the actionable RNA biomarkers are mainly long RNAs. So a little bit about what that pipeline looks like and how it can be used. So it's been designed to accept inputs from things like plasma, serum, CSF, urine. Others are possible with development, like saliva and ascites fluid. I'll mention here also, when thinking about leveraging us as a partner for your programs, we also can run total tissue RNA from things like FFPE or fresh frozen tissue in the same pipeline. We would do a total extraction and not one of our proprietary column extractions, which isolate high quality, high quantity exosomal RNA. But the library prep itself is actually very amenable to cellular RNA modalities as well. And we have a number of panels as well as whole transcriptome library approaches that we could apply to any of your programs. I'll focus on our whole transcriptome today. And what's most exciting for us and for our partners is that this provides similar diversity and detection to what you see with tissue. And so if you're familiar with any kind of tissue sequencing, you know you're going to see a lot of protein coding genes. This chart here on the right is capturing the number of genes detected across these RNA biotypes. And while this may be challenging to see because the words are really small, hopefully you can appreciate that's because you actually capture a wide diversity of biotypes captured in these exosomal cargoes. And the biotype that is by and large the majority are protein coding mRNAs. And you'll see other things like long non-coding RNAs, which are very relevant and interesting to researchers. This chart here also demonstrates the detection that you get from either 2 mLs being the light blue or from ½ ml being the dark blue of input plasma. And so for this study, a 2 ml aliquot and a ½ ml aliquot was extracted through our exolution column, subjected to our whole transcriptome proprietary RNA-seq process and analyzed. And hopefully you can appreciate that it's very difficult to discriminate the data that comes from either 2 mLs or ½ ml. And that's really because you get the same diversity of detection, whether using 2 mLs or as little as ½ ml, which is very empowering for our partners' programs. Very little plasma is needed to do a lot of work. And so what kind of data, what kind of information can you expect from a typical exosomal RNA sample? So the Venn diagram on the left is capturing gene detection and essentially using as input either CSF, plasma, serum, or urine. And so the key part here is across these 4 different biofluids, we can use the same core technology and then of course the same RNA-seq preparation and analysis pipeline, and then compare the genes that are detected from these different biofluids. And importantly, these are in the normal sample setting as well. And so hopefully you can appreciate that the largest number is shared by all these different biotypes, but there are still unique genes that are detected in those biofluid compartments. And so what that really says is that there are many transcripts that are observed across one or more of those fluids, but there are often things that are unique. So when we have partners that come to us and say, I'm working in plasma, but I want to try it in urine, we often can say there's a very good chance that will that your gene will be detected in plasma or urine, but we won't know because there are still things that are unique. So we will always investigate to see, but there's a very good chance. And so gene detection is only part of the story. The other is the gene expression profile. And so that's what this PCA plot here on the right is demonstrating. And so the structure and where these biofluids separate to or overlap is driven by the expression of these genes that are detected here. So not surprisingly, plasma and serum essentially being the same or very similar biofluid compartment overlap as you might anticipate, whereas CSF and urine separate from blood. And so what do we do with these with the patterns that we see, with the biomarkers that we identify when using the RNA-seq pipeline, we would then take that focus and make a targeted approach. For example, here, this example is targeting mutations and variants that are prevalent in lung cancer. And we would then use, for example, qPCR to target and refine for a translational or a clinical assay. We could also apply other platforms. And so I won't go into the specifics of what was the methodology for deciding what the targets were going to be, but needless to say, this is a qPCR assay targeting prevalent fusion variants that capture a majority of lung cancer variants. And this is an illustration of what a very typical assay workflow for an exosomal sample would look like. Anywhere from 1 to 4 mLs of plasma, so very amenable, very low input needs through our proprietary extraction technology, RT and qPCR for targets of interest, leading to a diagnostic result. And basically, as you might expect to see here, where you're looking for ALK positive, here we did a comparison against samples that were orthogonally validated by tissue fish status to have ALK rearrangements, and then we can detect those ALK rearrangements as positive ALK readouts in those plasma samples as well. And so one of the things that's very new and super exciting for us at Exosome is a recent partnership that we press released with QIAGEN. And so we've had a relationship running with QIAGEN for many years, but we've just expanded upon that. And it's very exciting for both of us to be able to be doing that. And so what does that mean to you as partners with Bio-Techne and Exosome? And what that really means is it's enabling QIAGEN's global scale and reimbursement experience, as well as their facilities and their experience in sample extraction, as well as all these other expertises that we share with them to quickly develop and support exosome-based liquid biopsy for your CDX needs. And before I hand the microphone over to my colleague, Dr. Chris Bunker, I'll just mention briefly that while I focused on things that were around qPCR or our NGS RNA-seq, we at Exosome do a lot of different exosome-based research programs, including supporting other Bio-Techne platforms. And so with that, I will gladly turn this over to Dr. Chris Bunker.
Hello, my name is Chris Bunker, and today I will speak about the RNAscope in situ hybridization assay technology of Advanced Cell Diagnostics, which is a brand of Bio-Techne. Like the Exosome Diagnostics platform that you just heard about, RNAscope is another way in which to expand the options and means of biomarker analysis and potential companion diagnostics. The RNAscope is an in situ hybridization biomarker assay platform that, like PCR, it has very high levels of specificity and sensitivity for mRNA detection. Unlike PCR, it works on routine formalin-fixed paraffin-bedded tissues. That makes it similar to IHC, and it has a similar workflow. However, in this case, we're hybridizing with oligonucleotides that are sequence-specific for a biomarker mRNA, and signal amplification occurs locally so that the signal that we detect corresponds to individual molecules of mRNA, which can be quantified with spatial resolution. RNAscope is a very robust method for mRNA biomarker detection in fixed tissues because of the probe design. As mentioned, signal amplification is dependent on pairs of oligonucleotides hybridizing immediately adjacent to each other to short sequences, about 40 to 50 nucleotides. Each pair enables signal amplification. So like PCR, signal is dependent on pairs of oligonucleotides hybridizing, but in this case, we use a very large number of oligos tiled along a region of the target mRNA. However, we only need a few of these pairs to bind to generate detectable signal. This built-in redundancy is what makes it so effective and reliable in detecting RNA biomarkers in fixed tissues. Advanced Cell Diagnostics has two in situ hybridization assay technologies. One is RNAscope, the other is BASESCOPE. RNAscope is the perhaps more robust assay in that it's been in use for about 10 years now. It enables higher levels of plexing, 3-4 plex assays on formalin fixed tissues and even higher plex assays on frozen tissues. It is also actually being used for diagnostic purposes currently, in particular for HPV detection in head and neck cancer. BASESCOPE is an assay which we use for specific applications, specific biomarkers, where RNAscope would not be suitable or not able to differentiate the intended target from non-targets. In particular, BASESCOPE is used for detection of splice variants, for instance, FGFR 2B and 2C variants. It can be used to look at fusion junctions. It can also detect specifically target sequences that are members of highly complex gene families. Moreover, it can be used for point mutation detection, and I'll show illustrations of that as well. This slide is from the first publication with the RNAscope assay, and simply is to demonstrate the fact that what we detect with RNAscope as dots, visual dots, correspond to individual molecules of RNA. This is a simple cell culture experiment where HeLa cells were tested with an RNAscope probe to HER2. On the left-hand side, you can see visual green dots, and there are approximately 14 dots per cell in a field of view. In comparison, another plate of HeLa cells had RNA extracted and measured in solution with a standard curve of in vitro transcribed message. In that way, an average value of 17 copies of HER2 RNA could be measured on a per cell basis. And so these two numbers, 17 copies per cell on average from a lysate and 14 dots per cell, are very similar. And this is to show that an individual molecule is being detected when we see dots in a fixed cell or fixed tissue. This assay was used by the lab of the late Ray Tubbs at the Cleveland Clinic as a means of differentiating or resolving, I should say, cases where HER2 status was equivocal based on either IHC or DNA FISH. What the lab of Ray Tubbs did was to measure HER2 transcript level in breast cancer and determine a molecular threshold that corresponded to HER2 positivity in comparison to IHC. This is a bit of a busy figure, but the primary point here is that using a molecular cutoff of HER2 measured by RNAscope, in the first place, they can show very good concordance between cases where IHC is equivocal, but FISH is negative for HER2 amplification, and RNAscope signal is also negative. In the middle, you're seeing cases where IHC again is equivocal, but it's FISH positive for amplification of the HER2 locus, and likewise, HER2 transcript levels are also amplified above that molecular threshold. The important data set was in cases where both IHC and FISH were equivocal and could not firmly establish the role of HER2 in breast cancer. In this case, what's shown is the same molecular cutoff can be used to define a patient as being positive above the threshold or negative below that threshold. So this is just a point about the ability to use molecular quantification of a transcript, in this case HER2, to define a patient as being positive or negative and potentially responsive to a targeted therapeutic. This and the next few slides will show examples where RNAscope was determined to be more sensitive and specific than other methods for measuring specific biomarkers that were predictive or appeared to be predictive of patient response to an oncology targeted therapeutic. The example shown here is for MERRIMACK Pharmaceuticals' MM121 anti-ERB3 therapeutic, where they determined that it was actually the ligand growth factor, heregulin, or NRG1, which was predictive or at least correlated with response to their therapeutic MM121. Now, the fact that NRG1 is a growth factor and secreted protein made it very difficult to detect it by IHC in intact tissue biopsies, and the fact that they were getting very small needle biopsies from the patient did not provide them with sufficient material to get a reliable qPCR readout. Moreover, the spatial resolution of the RNAscope assay enabled them to verify the presence of heregulin expression in the tumor itself as opposed to in adjacent stromal cells. And these factors contributed to its utility as a potential biomarker assay. Shown here is retrospective data from phase 1 clinical trials in breast cancer, ovarian cancer, and lung cancer, where they observed that high levels of heregulin as measured by the RNAscope assay appeared to identify a subpopulation of patients which were responsive and showed longer progression-free survival in comparison to patients with low heregulin expression. It was based on these results that they used this assay as a basis for patient selection in a phase 2 clinical trial. RNAscope also proved to be a very effective biomarker assay for BAYER'S FGFR 1, 2, and 3 small molecule tyrosine kinase inhibitor. In this case, it showed superior performance compared to IHC and nanostring. And the starting point for this was the fact that historically, gene amplification and genomic alterations of FGFR genes has been used as a way to identify patients for small molecule tyrosine kinase inhibitors. However, published reports showed that there actually was not a very good correlation between TKI sensitivity and gene amplification, and in fact, expression was a better predictor. And based on these results, they used the RNAscope assay as a basis for enrollment in their phase 1 clinical trial, as well as their phase 2 clinical trials. And in both cases, both clinical trials, there was a very high rate of response to their tyrosine kinase inhibitor in patients that were selected with the RNAscope assay. And this is just in one illustration here, a case where there was no genomic alteration in this patient's biopsy. However, there was a significant increase in FGFR RNA expression, and this patient did in fact respond very well to treatment. This was observed in a very large percentage of the patients that were enrolled in this trial. The IDO1 checkpoint target is another good illustration of where methods like RNAscope can be more effective than more well-established approaches like IHC. In oncology, IHC is probably the default method that people think about when they think about a biomarker assay. However, oftentimes it will be the case that newer assays like RNAscope, as well as exosome diagnostic assays, may be more effective and will certainly be much more time efficient and cost effective to develop. I mention this headline here to just illustrate this point, which is that it was about 3 years of time spent by Insight with Roche Ventana to try to develop an IHC assay, which in the end was shown to be about 10 times less sensitive than the RNAscope assay for measurement of IDO1 expression. What's shown here is cell culture work on HeLa cells where interferon gamma can be used to induce IDO1 expression. And what was observed was that in a dose response to interferon gamma treatment, both Western blot and RNAscope were able to detect IDO1 expression at a 10 times lower dose of interferon gamma, indicating a greater sensitivity. So while RNA and protein correlate very well with one another, in an assay of intact cells or tissues, the RNAscope assay was proven to be more sensitive than IHC. And as a result of this, they went on to use RNAscope in collaboration with ACD in multiple phase 1 and phase 2 and phase 3 clinical trials, testing well over 2,500 patients. Again, it was unfortunate, certainly, that the drug proved not to be very efficacious. However, from a technical standpoint, the results showed that the RNAscope assay was a very robust and reliable method of biomarker analysis for this particular biomarker. Another example for an ongoing clinical trial with Leap Therapeutics, where their anti-DKK1 antibody is being tested in patients where DKK1 expression is being measured by RNAscope. Here, like in the case of MERRIMACK'S MM121 therapeutic, the target is a secreted protein, and so measurement of that by IHC is not very effective, whereas measuring the expression of the DKK1 transcript and identifying the cell populations expressing that is something which can be provided with the RNAscope assay. And here, like with heregulin, high levels of DKK1 expression identified a patient population with a higher probability of long-term survival compared to those patients that had a low DKK1 expression. So this is an ongoing clinical trial, phase 2 clinical trial, and could lead to companion diagnostic development. Here is an illustration of our BASESCOPE RNA in situ hybridization assay showing detection of EGFR point mutations, namely T790M as well as L858R. Now, in the previous slides, I showed examples of our RNAscope assay for measurement of expression biomarkers. And oftentimes with gene expression, particularly in the case of oncology, it's important to understand what cell types or regions within the tumor microenvironment are expressing that biomarker. And that's something which comes from in situ analysis, which gives you morphological context. Oftentimes in the case of mutations, the mutation itself is indicative or diagnostic of an oncogene being active. And that can be measured either by in situ hybridization as shown here, or by methods like the exosome diagnostic approach. And here, again, just illustrating the fact that we can in fact detect point mutations and measure point mutation expression in intact tissues. This type of assay could provide a bridge to a liquid biopsy assay such as that of Exosome DX, using the two assays to show correlation between analysis in tissue and analysis in a liquid biopsy. Likewise for ALK, or the anaplastic lymphoma kinase, as was shown in previous slides, fusions which are activating mutations of ALK can be measured by PCR from exosomes. Here we're showing measurement of overexpression of ALK by RNAscope, overexpression being the result of fusion to various different genes. So here again, like with EGFR and other mutations, it may be useful to use the RNAscope method and exosome analysis to cross-confirm and verify whether or not a liquid biopsy approach will be suitable. However, again, if there is a reason to think, or if there's data supporting the fact that there's information in spatial resolution or in morphological analysis of a mutation such as this or EGFR, that's where an RNAscope or BASESCOPE assay can be applied to provide spatial resolution of expression patterns or mutations. So our assays, like those of Exosome DX, Exosome DX has a partner in QIAGEN. We have a partner in Leica Biosystems for companion diagnostic development. This is enabled by the fact that assays being run in a research setting where biomarker assays can be developed work on their research use only instrument, their Bond RX, and those can be easily ported over to and validated for use in a diagnostic setting on their diagnostic Bond 3 instrument. We would make a case, obviously being somewhat biased towards the RNAscope assay, that there are many advantages and benefits to the RNAscope approach compared to other methodologies. In particular, we do emphasize the importance and value in analysis of biomarkers within the morphological context and having spatial resolution of what cell types and regions within a tissue expression is being observed. That's something which is obtained both from RNAscope and IHC, but not extraction-based methods like qPCR or NGS. The assay with RNAscope is oftentimes much more sensitive than similar tissue-based methods like IHC, has comparable sensitivity to extraction-based methods like NGS or qPCR. Likewise, specificity is very, very high with our molecular analysis with RNAscope and those other extraction-based methods. A benefit, a potential relevance to clinical analysis is the fact that the RNAscope assay requires very minimal tissue, just a few 5-micron sections. Oftentimes, methods that are based on extraction of nucleic acids require a larger amount of material. So overall, what we have to offer with the RNAscope assay as well as BASESCOPE are assays and probes that can be used to detect virtually any target or biomarker. The illustration with IDO1 in relation to the IHC assay that was attempted in that case is the fact that we can develop probes and assays very, very quickly. These assays can also be multiplexed with cell markers or multiple targets where that's relevant. And again, the benefit of a nucleic acid-based assay is that it is very highly sensitive and quantifiable on a per cell basis. In summary, what we have presented today are two biomarker platforms of the Bio-Techne Corporation. One, the Exosome platform of Exosome Diagnostics, and the other, the RNAscope platform of Advanced Cell Diagnostics. Both of these have advantages of very high specificity and sensitivity that are inherent to nucleic acid-based tests. In addition, the Exosome platform offers the advantage of being a liquid biopsy-based test, and the RNAscope platform offers the advantage of spatial resolution in the context of intact tissue morphology. Both assay platforms are easily implemented, and with our partnerships with QIAGEN in the case of Exosome Diagnostics and Leica Biosystems in the case of Advanced Cell Diagnostics, both have a straightforward and clear path for companion diagnostic development. With these factors in mind, we hope that you will consider inclusion of Exosome Diagnostics and RNAscope in your clinical biomarker strategies. Thank you very much.
Thank you, Dr. Monroe, Dr. Bunker, and Dr. Valentino for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, let's get started. Our first question asks, how are exosome liquid biopsy biomarkers validated as compared to solid tumor markers?
Thanks, Kylie. This is Mike Valentino from Exosome Diagnostics, and that's a great question. I think actually the way we would approach validation is really similar to how other people may be familiar with solid tumor marker validation. It's not all that different. And the way that we think about it is kind of twofold. So if in our very broad, for example, RNA-seq-based approach, you identify some biomarkers that are really interesting and relevant, but often you don't know what that's going to look like in a broader patient cohort, larger demographic, and really want to ask additional questions as to the utility and validity in the clinical sense for those biomarkers, we would develop a more targeted approach, whether it be NGS or qPCR-based panels, to take those preliminary biomarkers and validate them forward in terms of their utility and usability in your clinical space. And once you go through and you validate those markers and that holds up in your patient demographics, we then would actually validate the assay itself. And so there's a couple of ways that you look to do that, analytical validation. And we're well versed in doing that both for our own programs and for our programs that we run together with our partners. Basically starting in the one setting and then moving it into our CLIA diagnostics facilities in order to support the analytically validated clinical assays.
Great, thank you. Our next question here asks, can you look at DNA or proteins along with RNA?
Kaylee, this is Mike again. I'll dive in on this first. Exosomes are actually a really powerful, robust resource for liquid biopsy and for investigation. And so, as I kind of alluded to on the slides today, we focused on RNA, but we do other things as well, like proteins. So just like the RNA-based cargo within exosomes, there are also protein cargoes, both contained within and embedded on the surface of the exosomes. And so we have programs that are focusing on exosomal-based proteins. And DNA is another thing that can also be captured and carried along with the RNA cargo as well. And I didn't at all touch upon this today, but we at Exosome also have a proprietary column isolation approach that captures cell-free DNA. And this is basically analogous to any of the cell-free DNA approaches that others support, where the DNA fragments are actually not contained within exosomes. These are actually free-floating, soluble DNA fragments. And so we have a column approach that can capture with the same performance as other gold standard cfDNA kits, RNA from exosomes, as well as cfDNA in a single one-step isolation, which has been very, very powerful, especially for mutation detection.
This is Chris Bunker from ACD. I'll add just a brief point regarding detection of various types of molecules in the context of RNAscope assays. We actually do a great deal of work combining IHC or immunofluorescence with RNAscope, oftentimes using either IHC or immunofluorescence for cell marker detection, in particular immune cell markers. So a combination of protein-based detection alongside RNAscope for mRNA detection, that's something we do routinely. We are developing methods, so-called DNAscope methods for doing DNA detection similar to RNAscope, but those will likely be completely separate assays from RNA detection.
Awesome. All right, we'll go to our next question here, which asks, what are some applications that can be or have been supported beyond oncology biomarkers?
Maybe I'll start with that. This is, again, Chris Bunker. Actually, a really important area for us in use of the RNAscope assay is in looking at kind of more pharmacodynamic or pharmacokinetic markers. Again, in the context of immuno-oncology, we use RNAscope probes to detect engineered cell therapeutics, CAR T cells and TCR T cells, by targeting viral sequences that are present in most of those cell therapeutics. And so that provides us with a means of monitoring and quantifying the infiltration into a tumor of cell therapeutics like CAR T cells. And likewise, oftentimes we combine with those assays or other immuno-oncology related assays the measurement of cytokines and chemokines, like the assays I showed for looking at DKK1 and Regulin, assays of cytokines and other secreted factors that are indicators of immune cell activity are very effectively performed with RNAscope, where we're looking at the RNA and the cell type producing that RNA.
Yeah, that's great, Chris. I think what is awesome between the ACD RNAscope technology and what we at Exosome can do, a lot of what Chris just covered is really how we think about it at Exosome as well, how we apply it in our programs, and kind of one of the main things for us in a liquid biopsy-based approach compared to other modalities for liquid biopsy investigation is that because we're looking at these RNA profiles, you're able to capture exosomes in both normal and disease settings, and we're not tracking only mutant molecules like you would be kind of stuck with other CTC-based approaches or ctDNA-based approaches, where you're only tracking mutant molecules as signals for your disease. And so we span well beyond oncology. We have active programs in neuro and many other IO approaches as well, really owing to the fact that it's RNA and tracking RNA expression differences and not really just built on polymorphisms and mutant tracking.
Great. All right. It looks like we have time for a few more questions. This next one says, how can exosome and RNAscope assays be used in pharmacodynamic studies?
I guess I already sort of answered that one. So maybe Mike, you can add to that.
Yeah, and I think again, it's very similar, and this is why they're really two great and complementary approaches. I think is really, I think the way Chris captured it is kind of how we would do, but also just in a liquid biopsy based setting.
Great. All right. So we have time for one last question here, and this one says, how do you validate single copy detection?
I'll sort of dive in on this. So single copy detection and validating in an analytical sense is can often be challenging. But when we strive to analytically validate or even just benchmark performance, we often will take advantage of samples that are available either from sample repository sources or anything that you guys, that our partners may have in their biobanks that we can take advantage of to explore for detection. But also we take advantage of synthetic molecules, either at the RNA level or the DNA level, and we can do spike-in experiments and recovery experiments into complex matrices like plasma, serum, CSF, urine, any biofluid that you're looking at, to be able to, within the complexities of your matrix of interest, really get a refined performance specification of the assays that we design.
In the case of RNAscope, to a similar extent, we rely on comparison to methods like qPCR or RNA sequencing, and also as well as nanostring assays to show concordance between what we can quantify in intact tissue by counting dots with what can be counted in an extracted sample, where it's easier to use spike-ins, as Mike mentioned, and other ways to establish a standard curve. And many times over, very reproducibly, we can show the ability to detect and quantify individual molecules of nucleic acid, in particular mRNA.
Great. All right. Well, I want to remind our audience that those with questions who we were unable to answer today, that those come in during the on-demand period will be answered by Nicholas via the email address you provided at the time of registration. So thank you again, Dr. Monroe, Dr. Bunker, and Dr. Valentino. Do you have any final comments for our audience?
No final comments. Just wanted to thank everyone for joining us today. Please feel free to reach out with any questions about the technologies or your interest in companion diagnostic development with Exosome Diagnostics or Advanced Cell Diagnostics. Thank you very much.
All right. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we do not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to once again thank Dr. Monroe, Dr. Bunker, and Dr. Valentino for their time today and their important research. We would also like to thank LabRoots and our sponsor, Advanced Cell Diagnostics, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time. Goodbye.